The journey to the center of the cell. The big room was crowded. I had no idea of where to go. Where could it be? Said it would be here somewhere. See, today was the day that I was supposed to start working for my new cell. One of the hundreds and hundreds of muscle cells in this leg of the creature that I am in. I have no idea where I am or where I'm going. The only thing I know is that I'm in a leg of something that walks on two legs upright and is really, really, really big. It looks kind of like the scientists who made me and put me here. They said I was supposed to make the cell work faster, the muscle move faster, and maybe even make the creature faster. At least that is what they hope. It is also the reason my name is Celeritate. It is Latin and means speed. At least that is what it said. Hello there, I heard someone shout. I looked around. Was he talking to me? Yes, you. Oh, he was talking to me. Maybe he could help me. Hi, I said slowly. My name is Celeritate. I'm kind of lost. I was sent here, but I can't find the right cell. My task is to make this an organism faster. Do you have any idea of where it could be? Yes, I do. I know exactly where you should be. You're in the right place. What was he talking about? Where is it then? Welcome, he said, to your new home. My name is Nucleus, and I am the brain of my cell. Your new home. Oh, is this it? How lovely! Would you like to take a look inside? I will show you around. Okay, so the first thing that a new substance or new organelle, like you, encounters is the cell membrane. This is what surrounds the cell and controls what comes in and out. It is the postcard of our home. Hello, membrane. I'm the new organelle Celeritate. Hi, nice to meet you. I wished I could talk, but I have loads of work to do. I'm the guard, you know. I understand. It looks pretty busy, I said and turned to Nucleus. How does it work? The membrane is made of the proteins, the long strings you see here, and phospholipids. Lipids are substances that have one hydrophilic end and one hydrophobic end. It means that one end mixes well with water and one doesn't. The way it is arranged with the hydrophobic, which is the not mixing end, to the center and hydrophilic outwards is called double bilayer. Understand? Yes, but what about the proteins then? Okay, the embedded proteins are, as you can see, twisted around like a tube. They work as channels for the substances entering the cell, carrying them in. Each protein has its own unique shape, fitting its own specific substance. Would you like to enter? Yes, I would love to, I said, and a second after I was inside. Next, there is the cytoplasm and the cytoskeleton. The cytoplasm is this thick fluid that fills the cell. It is a mixture of water, dissolved salts, ions, and organic molecules. It feels weird, I said to the thick and sticky fluid. Suddenly, I felt something grab a hold of me. It was a long thing that had what looked like legs. Ah, what is this? I screamed. Oh, the Nucleus chuckled. This is the motor protein that will move you around on this guide. And what it walks on is the cytoskeleton. Cytoskeletons are what keeps everything in place and forms the cell. They are the bones and muscles of the cell. They shape it, anchors organelles, and direct movements. That is so cool, I exclaimed and looked around. It looked like the busiest cities that I saw in a box called television. There were huge organelles like buildings and long cytoskeletons resembling roads. I was overwhelmed. Do you see these small balls going around the thin strings? I nodded. There were actually many small clamps working around the thin strings. Tiny things flew in and out of the ball as it worked around. Also, there were proteins forming and coming out the top. When the clamp had circled one time, it broke apart and released the protein and the string. These are ribosomes, the nucleus started, the smallest organelle in here. They are made of long chains of proteins molecules. The thin chains did you see, are messenger RNA or mRNA, which are copies of the DNA, which is the genetic information found in the nucleus. The DNA are made up of nitrogen bases. Triples of these nitrogen bases make up an amino acid, which is the buildup of proteins. The mRNA is scanned by the ribosome and a protein is built. Then the ribosomes break in half and release the protein and the mRNA. Protein is then carried to the place where it is needed. I would like to go into more detail, but I cannot. I see. This was a lot of information. What is this? I exclaimed to the folded membrane surrounding the nucleus. This is the endoplasmic reticulum. What a long name. What is it for? I asked. The endoplasmic reticulum, or ER, is made of the same type of membrane as the cell membrane, but here it is folded and divided into two parts, the smooth and the rough. The rough ear is what is closest to me. It is called rough because of its surface. Can you see what it is? Oh, it is covered with ribosomes. That is totally right. These ribosomes are called bound ribosomes. The proteins that they make are stored inside the ER to keep them separated before sending them away. The membrane is produced by the rough ER itself, which is important, since there needs to be something filling in after the transport vesicles. The sacs, that you see, are pinched off from the membrane to take the proteins away. You see them? Yes, I do. What about the smooth ER then? The functions of the smooth ER can vary depending on the function of the cell. As you know, our cell is a muscle cell, and therefore the function of our smooth ER is to produce and release calcium ions, which in turn triggers the contraction of muscle cells. Now let's follow the vesicles to our next destination. Oh dear, Golgi apparatus. This is where the vesicles are transported. This too is made of membrane, the same as the ER and the cell membrane. The ER sends its transport vesicles containing the proteins here where they are modified and stored. When they are ready, the Golgi apparatus sends them to where they are needed, which could be either inside or outside of the cell. Wow, I exclaimed. It is all connected. Hello, Golgi. I'm the new organelle Celeritate. You're doing great. Oh, hello and welcome, he replied. I hope you like it here. I sure do, I replied to Goldie. It is very nice. It is, Goldie replied, as he collected some small balls that looked kind of like the vesicles we saw earlier. What are those? I asked and turned to Nucleus. They are called lysosomes. Can you guess what they are made of, Celerate? Maybe membrane? said with a giggle. Yes, you're completely right. These are, as many of our other friends, made out of membrane, the same as what surrounds us. As the vesicles, these small balls, are made in and transported from the rough air, but instead of carrying proteins, they contain enzymes, which digest macromolecules so that they can be used in the cell. Every different lysosome has its own special enzyme that breaks down its own specific molecule, and every molecule produced from the breakdown is very important to the cell. Here comes some more, I said, but before I got to say hello, they were inside. As we continued, I saw another of the membrane sac. It looked just like the vesicles, but much bigger. Wow, this looks almost like a vesicle. Is it possible that it is made out of membrane? Yes, it is. You're starting to get this, little one. This particular membrane sac is a vacuole. Vacuoles are mainly used for storage of different kinds. This, for example, is a food vacuole. It stores, as you might understand, food and nutrients. Well, that is good, I agreed. Hi, vacuole. I'm the new organelle, Celerate. Oh, nice to meet you, he replied. I hope you like it here. I sure do. Now to our last but yet so important organelle for this trip, the mitochondria. The nucleus explained as we continued through the cytoplasm of the cell. There were many of them, maybe hundreds, and all of them worked like crazy. The mitochondria are made out of membrane too, he explained. The membrane is in two layers, one outer layer that is smooth, and an inner layer which, as you can see, has folds. These folds are called cristae, and they increase the surface area of the mitochondria for all the reactions that occur. What reactions? I asked, confused. Well, the mitochondria are our factories. They take nutrients from the humus foods and break them down in order to release energy for us to use. This is called cellular respiration, and it is crucial for our survival. Wow, they must be very important. They sure are, and that is why they are able to reproduce by themselves. See, mitochondria have their very own DNA, which is their genetic information. Because of this, they can divide and reproduce in case there are too few. Oh, you're doing great, mitochondria, I said. Thank you, it replied. You're the new cell, right? Welcome. Well, Celerate, that was it. I truly hope that you will enjoy your new life here. It is a pleasure to have you, and I wish you a good start in this lovely little muscle cell. Have fun and work hard. Thank you so much for welcoming me in such a nice way. It was so nice to meet everybody here, and I think I will enjoy my stay. It is a wonderful place filled with so many wonderful organelles, who all are so incredibly important for our survival. Thanks for the journey to the center of the cell.